above them? See above
Yeah,
them?
how much time? We have about a little over 2 hours left.
That's right. So what do you think? We got a super from Mike. Tara Luke agrees with Deb, who says a white rock sample would be great. And David Butterfield says choose wisely.
I just smushed my skid and I actually think it looks a little bit like clay.
Ah.
If I back up,
Into
I
the
can
white
show you.
material?
Yeah. See, look over on the left-hand side.
Actually, I thought
Okay.
it looked like clay from the beach at home when I came up.
Okay, folks, take a look at the left side of your screen. Now, the pilot just kind of mushed the
Okay, video, you
sled
want to zoom in?
into the material. So this looks like, it's very soft, easily mushed material. It does look like
sure
clay.
Does look like clay.
Yeah, when we were drilling on DSTP Lake 60, we did penetrate some marley, carbonate-rich, but nanofossil and foraminiferal material that had a lot of clay in it. This might be altered ash.
Come out a little.
It could be an ash layer, really heavily altered. Not sure what the stuff is, but I'm guessing that if we tried to pick up a piece of this white material, it might just crumble in the manipulator. But up to you as to whether we give it a try.
I think it's going to be hard to collect.
I do too.
I think it's going to be very hard to collect. I think we should probably move on.
Yeah. Okay, Deb says, totally looks like clay. Okay. This is a no collect.
All right.
So, pilot, thank you so much for jamming the ROV into the side of this thing so we can figure out what it is. But that's really good to know. Thank you.
Welcome. Now, if you want to drop a target, oh yes, you can go wide video. Now, I want to drop a target clay.
Okay.
All right.
Probably, do you have a visual on my sonar return there?
Mush looks like clay.
Okay. How far away is that? Is that me?
Looks like you're dead ahead of me, right?
Yeah. And then you got some more. So after you're officially stopped here, we might want to think about turning you a little to port to follow this ridge up.
Yeah.
You can see it in my screen too. It's veering off that way.
I think we're settled out now.
So?
Yeah. Navigator,
well, there is a
when
lot
did
of
we
this
stop?
stuff.
Yeah, there sure is.
We stopped at 53.
Yeah.
Well, it is a volcanic arc.
Just under 10
If it were
minutes ago.
clay,
Okay.
altered from volcanic ash, particularly
Alright,
if
do
it
you
was a
wanna,
silicious ash,
Kind of bring your heading around.
it could
to
look like this.
follow this ridge.
Yeah, let's see what we can find here.
Oh my.
Yeah. Based on your
The
sonar,
series
I'd say
view
look at
is
the view. Oh, gosh.
spectacular,
Missed that.
folks.
Yeah.
As
Was
we
that screen
Yeah,
two?
well,
Was
they're
that
screen,
screen
They're looking at
your
the view
feed
now, you
two,
can keep it.
the center one in the bottom of
But
the
yeah.
3 on the bottom of your
Monitor.
web page
Something like that.
shows the view from the Sirius vehicle, which is attached on that 30-meter tether to the remotely
Yeah, you
operated
like that
vehicle
heading? If you did
D2.
30 meters there, you'd end up right on top
Gorgeous
of it.
view of the drop-off
That'd
on
be
the
good.
edge of
Yeah.
this little
All right, Nav.
sort
Let's
of
do a move.
lightly sloping.
Sounds good.
We're faced
ridge that we're sitting
240
on.
now.
Looks pretty steep, huh?
Well, that part sure was steep. Yeah, the ridge, the flat
The ridge
part.
is very- yeah, it's very
but that
gradual.
is
It's like a-
dipping steeply. Well, not
Yeah.
steeply, but is dipping up slope. So we're still moving up
Okay.
slope in front of the ROV.
Actually, we could do.
We're at
I think we could do
4,313
'cause
meters.
we're doing
Right.
0.1, right?
And about 2 meters above the
It
seafloor.
looks really clear ahead of you, I think.
And the
Yeah, I'm
water
happy.
is crystal
Okay.
clear down here.
Yeah, well, let's see,
So,
see how
like
the ship
Scott-
can handle the
France said,
move.
so
So,
not a lot of
40 meters,
particulates,
bearing 240.
so maybe not enough
speed
food
0.1.
for very
I'll
many
call it.
critters down here.
Bridge from Nav.
Just gonna peek down the edge.
Hello, bridge. Could I have another move, please? That would be 40 meters, bearing 240, and a speed of 0.1 knots. Good copy, Bridge. Thank you.
I pulled up just a little bit.
Okay. Video, I think I see a shrimp here.
Ooh, a shrimp? Let's take a look.
Alright, I'm gonna get one skid in here.
Oh, yep, I see it.
Okay, you're free to zoom.
Biology.
Yeah, it seems like the shrimp are the
Go ahead,
most
Nav.
common
Pack
meter
is defaulted to
critters.
feet.
Okay, so on the
This
top,
looks like the one we
to
saw
the
before.
It does.
Yeah.
Yeah,
Unit,
it does. Sure does.
Okay, I'm
Oh,
gonna
I
try
like
to
this
go
little,
right.
where it says
Long
U-
antennae.
USB.
Long antennae, and you can see the little blue,
Up a little bit.
Okay, you can go in.
whatever they are,
Up.
hairs that are coming
Right,
off its.
Right, a little bit. Yeah, that one.
some of
the
Clicks
antennae.
that. For some reason, when you
Antennae,
first click into high pack, the first thing it
Feelers.
selects
That's
is, yeah. So if you hit the arrow keys and you're changing your units, you don't know. That's why I like to click in the map somewhere.
Oh, look at the
So
little
that way
feet
I'm
in
sure,
front,
yeah.
waving in
It's
the
a high
water,
pack thing.
maintaining his position, no doubt.
Mhm.
Or maybe grabbing
Scavenging,
for food.
right?
Yeah.
Scavenging for food off the bottom.
Mm-hmm.
Taking advantage of any food source it can get on. Oh yeah, you can see the little fine hairs on
Yes.
the antennae now.
And when they move those
It kinda like-
antennae against the light, sometimes
Yeah,
it
the way the
looks
light hits it, it's
shimmering.
shimmery. Yeah, like kind of blue.
Right, really pretty.
Very pretty.
Okay, whenever you're clear, video.
And still, the surface of the rock's just covered with this sort of teeny little hairs. It has to be something biological. Has to be, right?
You mean on the shrimp?
No, on the rocks.
Oh, on the rocks. I was like, oh,
Every rock has this stuff on it.
yeah.
Wow, look at that very narrow little chute.
Grooves, those grooves in the
Yeah.
rock.
I think whatever the rocks are, they're heavily altered.
Co-pilot, you think you're moving yet?
Not quite.
Okay.
So, Chris Kelly, are these shrimp adapted to eat both by scavenging along the bottom and also by capturing particles from the water column?
Boy, I have no idea.
Ooh,
It's got
white
pretty
rock up there.
fine pincers there, her legs, if you will. Something's going on with a couple of those front legs, too. He's presumably eating something really, really
25.
small. And that's about all I can say.
And does anybody know, there was a question earlier about whether or not the polychaetes that we saw in the water column, if they actually could creep along the bottom and scavenge along the bottom for food, as well as capturing particles out of the water column. Does anybody know the answer to that?
I'm not a worm guy. By the way, did you see the movie Godzilla? That's what that's in reference to. But never mind, stupid joke.
Yet again, Chris?
Yeah,
Video
maybe.
pilot.
What a shock.
Yep, video here.
Let's zoom on this yellow.
Okay.
Okay, I'm going to go ahead and Google this and see if I can find something out. I bet Scott knows the answer.
I think Scott is making his dinner.
Hi, this is Nolan at Harbor Branch. I don't think they could be used for crawling along the sediment just because the CD of the polychaetes look like they're
There's
specialized
something
for
in there.
swimming
Can
in the
you
water
go back
column.
soon?
Thank you, Nolan. So that is Nolan Barrett, and Nolan is a summer intern who's actually working in our drug discovery program this summer
Coming up
at Harbor
slowly.
Branch, but he's been a regular
worm to the
in our
right,
Exploration Command Center. Thank you very much, Nolan.
Just below the screen. Just below the
We're-
screen on the right. Sorry.
Tracking a little clear one in there.
Okay. Okay.
And a little bit more zoom
So
to go.
small. Yeah, can you go in coming up?
Our videographer thinks he sees some critter that we are looking for in the water. Oh, yes, swimming along. It's very tiny, folks. It's kind of, oops, just went off
Oh.
the top. It's kind of hard to keep track of these things.
Okay, let's go wide.
Looked like a little
That was so small and far away.
amphipod to me, kind of translucent.
So watching, did you say there was a worm in here somewhere?
Oh, I thought I saw something.
Down at the bottom, maybe.
I
Yeah.
don't know where it is
Yeah, I think
now.
we can keep
Sorry.
going.
Okay.
Yeah, we can keep going.
Keep on a going.
Is that full wide video?
Yep, that's full wide.
Thank you.
A lot of rocks sitting up on this ridge.
How's progress, co-pilot?
Might just be starting to move, but not 100% sure yet. It is a slow move.
Okay.
Yeah,
Judging
look
by
at
the
that
ship's
groove right
track,
there.
it hasn't-
We're just
Mm-hmm.
They're bringing their heading around a lot, so
Yeah, true that. Yeah, it's gonna be hard to. Okay. Yeah. I gotcha. And I think we're starting to move.
yeah.
Cool.
Okay, video. Let's go in on this little fish.
Oh, fish. I want to see the fish.
Are lasers on, co-pilot?
Yes, they are.
Wow.
Oh yeah, I see that.
That's another one just like the other one.
yes, it is.
It's another one of these
Okay, you can go in,
little white guys.
frame them up.
Oh, and it's turning around so beautifully for us. A beauty shot.
Okay, you can frame it up.
It's a little gray patch on the top of its head.
It's all yours, video. I'll
Big
just try to keep it
Yeah,
straight.
Bruce Monday remarked earlier in the chat log that the eyes look like something prehistoric. The absence of a lens makes them look like dark pools, and it gives the fish a death-like stare. It's kind of a spooky gaze.
But this is fantastic
Yeah.
video. This high definition imagery is so wonderful for the biologist. Shirley's been telling me for some animals, because the video is so clear and we get such close-up views that even the animals could be even identified down to the species level. This really impresses me. I wish we could do that with the rocks.
This is Bruce. If they can zoom, well, the fish is turning away now, I guess. Never mind. I was going to ask if they could zoom in on the head even more, but it's too late, I think. The presence or absence of scales on the top of the head around the snout area is one of the things that
You
helps
wanna get the
separate the
top of
two
his head?
species. The ones that we saw previously in the Marianas on the
Okay.
last leg
Go
were
back some video.
Leucochorus atlanticus. The ones that we saw in
Is that it?
Hawaii
That's it.
were
Okay.
identified.
Bruce is looking
along
for
the edge of
scales
the
on the head.
The genus as
Just let 'em
Leucochorus
swim
okeosus based on the absence of scales on the head. but we still might be able to see that from some images from these videos. One interesting thing is that this genus seems to be the most commonly seen
Is it top of his head or side of his head?
at these depths of 4,000
top of his head.
meters on these dives. And yet they were previously unknown from these areas in the Marianas and the Hawaiian Islands for the Okeanos Explorer expeditions. A lot of the observations of these fish and others from these extreme depths of 4,000, 5,000 meters and below are from baited camera systems that have been dropped down to the water. This species, or this genus, feeds on polychaetes and small crustaceans, and unlike some of the larger cuspioles or some of the rat tails, it doesn't seem to be a
profile view. There you go.
So it's probably not attracted to those drop cameras with the bait. So it wasn't seen by the systems that somebody like Jeff Drazen uses, for example, to explore the deep sea. But we're getting images of these. And even though you couldn't say that they're abundant at these depths, they're one of the most frequently seen types of fish at these depths that we're at now. So it's kind of interesting to see the observations from the ROV of the Okeanos Explorer give us new information about things that aren't seen or sampled by other types of gear used in the deep ocean.
Thank you, Bruce.
So we did get some video of it on the fly, and I would imagine when they do some screen grabs with this high-def imagery, you might be able to distinguish whether or not there are scales on the head.
Yeah, and thank you to the pilots and the video operators for that. That's great. These images are really wonderful, and the experts on these groups are really excited to see them because they're getting views of live fish that they've only seen from sort of beaten up specimens coming up from trawls before. And the images often are so good that they can come up with ideas about the species. So thank you again for the pilots and the videographers.
I'll pass along those kudos.
It's our pleasure.
Yeah, good. They heard you.
So it's not just the experts who can identify these organisms that benefit from these really high-definition images. I've been teaching a deep-sea biology class to undergraduates for 12 years.
Oh, yeah.
And up until very recently, the type of imagery that I could get to show my students to identify fish from the deep sea has been, well, you know what it looks like. It's old, it's grainy, or it's preserved specimens. And this is amazing. It's actually going to be able to allow my students to actually see what these fish really look like and maybe be able to tell the differences between them.
We are moving. I got you at 37
Yesterday I was
degrees,
looking at the literature for carnivorous sponges because I was trying to identify the 3 different species that we collected. And when I went and looked at the literature, the images there were all from dredged samples, so they were all macerated. So it didn't really help me very much. So I'm going to have to do, spiculoprobes, which I am not going to do because these things are really, really tiny. We don't have a compound scope, and I don't want to mess them up yet.
Okay.
So eventually we will do that.
With Sirius or the ship?
No, with the ship.
Okay.
Your Sirius view, which is
Makes a huge difference.
your number 2 feed at the bottom of the screen, shows the incredible steepness of some of the portions of the edge of this scarp that we are traveling up. And we are not even at the top of the scarp yet.
We have about 2 more hours left in the dive. I think that's
Right.
about 2 hours left, yeah.
You know, the thing that amazes me is you will map a big
this is
feature
your
like this
co-pilot here.
overnight
What's our
and get a
distance
really
to
good idea
waypoint
of the
2
general topography.
from,
And
D2?
then when we get down on the seafloor, I mean, every time we do it, there's so much more detail down here.
Yeah.
It's like a fractal exercise, you know.
Yeah, we had no idea looking at the multi-beam
original,
imagery.
yep.
Well, the multi-beam imagery was really the multi-beam imagery. The multi-beam imagery was really good, but
Yeah, but I'm
good.
thinking
It was
from
like
the original
from the
imagery we
original
had
to the satellite. Yeah, it's a multi-beam.
to being down here, it really is a fractal. Oh, God, exponential increase in
Up
detail.
a little bit, Aaron.
And then, of course, when you look at the rock surfaces, everything's all
You're definitely
broken
on
up
the
in
move.
tiny little fractures. Terribly complex. Incredibly confusing. And wondrous.
And do you have our off bottom time?
I think it's a late recovery today.
It's late, yeah. 50 meters at 6:30, right? Or
Yeah.
18:30.
Yep.
Right.
The original plan was around about 3:30.
3:30. Okay.
So we have about, let's see, 1:30, about 2 hours and 10 minutes, something like that. 2 hours and 10 minutes till we're off bottom.
3:30 seems a little early. 'Cause that would be 180 minutes.
Say again, Jim? I'm going to do a quick.
Well, folks, we're
training
coming
course.
to you live from the NOAA ship Okeanos Explorer, America's ship for ocean exploration. And we are in the Mariana Trench National Marine Monument. We're diving on a scarp that is reasonably close to the Mariana Trench. We're at a depth of 4,301 meters. And expeditions like this, the 2016 deepwater exploration of the
floating object
Marianas
in
is
my,
only
between
possible
me and you,
through
see that?
successful partnerships
Small
and
one?
collaborations.
Yeah, pretty small.
This
Your light
mission is
pool,
designed
I get a
and supported by
silhouette,
NOAA's
if that
Office
makes
of
sense.
Exploration and Research. The ship itself
Little
is
guy,
operated
float.
by NOAA's Office of Marine and Aviation Operations. So this expedition is part of a 3-year
He's
campaign
way closer
to
to me
address
than you. I think.
specific monument science, technology, and ocean needs. So the acronym for that is CAPSTONE. So you may hear us say that. It was designed to address fundamental need of resource managers to better
Still
understand deepest parts of the
indicating it's 3:30.
nation's marine monuments. Also,
Well, what is
the
the
fisheries,
The current depth of 4,
marine fisheries, and
wind speed minus 33.
really
I'd do
Oh.
32 maybe.
looking for the partners. We have a whole bunch of partners that
What do you
are
get for minutes
working
to
together
surface?
to bring you these expeditions. These include the management of the monument.
Yeah, so
as
we have
well
the-
as,
right.
the
Right.
National Center for Environment.
National
Perfect
Center for
there.
Environmental Information. So that's
Global
the-
Foundation for Ocean Exploration.
time,
These are the
estimated
people
time.
who actually are ROV operators, designers, builders.
right. So we have a number of NOAA partners. So, as
that
Patty
again?
mentioned, Ocean Exploration
Yeah,
and
minutes to
Research,
target
NOAA's
depth is
Office
what
of
you're
Ocean
looking for.
Exploration and Research is actually
Target depth
yeah.
is,
designed and
133.
planned
Okay,
this
that's about
mission
2
and
2 hours,
staffing
so
it.
we
But
would
in
be bottom
addition
at
to that,
4:30 then.
we have partners from NOAA's National Marine Fisheries Service and NOAA's Deep Sea Coral Research and Technology Program, who are also on board the ship. And
So we
let's see,
have-
who
yeah,
am I
we have
forgetting?
3 hours
National
on bottom
Monuments,
then,
the
right?
Monuments
Yep.
Program, and then and in addition to
Number
the global
of academic
number of academic institutions
Hawaii,
as well. Yeah.
Click
University of
enable
Hawaii.
edit on that yellow bar at the top of.
We have
yeah.
people from the
That
University of
Yeah.
Guam Marine Lab who are
Then,
involved.
maybe
Yeah,
edit
Florida
one of the
Atlantic
numbers
University.
there.
We've heard Deb at Harbor Branch, Florida Atlantic University, Deb and Nolan. And then all of our science team who are participating in this call today. So,
fish.
University of Hawaii. Okay, I'm going to get this wrong. We have University of Louisiana and Louisiana State University. And I'm not going to say who's working half-time where and half-time the other place.
Okay, when I get his name here, see what it is.
Let's see, who else? We've got Stockton University. We've got the P.P. Shearshoff Institute in Moscow. We've got the Chiba Institute of Technology in Japan. Let me see who else.
And we have a shrimp.
I know I'm forgetting somebody.
I think
4,000 meters.
this is
want me
the
to
same sort
get
of shrimp
hydraulics
we just
going around?
saw. But from this angle, it looks like it has a lump
keep
on
warm.
its back.
University of Washington.
Oh, University of Washington.
Oh,
Sorry about
there.
that.
Kind of too late now, but
Oh, go.
This
yes.
shrimp has a very strange morphology, Shirley. What is going on with this
trying to
shrimp?
kind of float
I
with
think
him.
it has a parasite on its back.
Well, let's.
What is that?
It's actually pretty fast for a shrimp.
Or
Yeah.
an egg sac? Who knows?
No, the eggs would be underneath.
Underneath?
Yeah, the eggs would be underneath.
Well,
Looks like it might be just something else along for the ride or a parasite.
a pillion to those of you who ride on
Still don't really make
motorcycles.
sense.
Well,
It's a much
Yeah, it looks like
lighter
looks
colored
like another.
shrimp.
Yeah, it
easy
looks
enough
like
to just,
another
add
type
the
of
numbers.
crustacean called a. Oh no, wait.
A mysid with
Yeah,
an
no.
isopod
Here we go.
parasite.
Yeah, with an isopod parasite.
Thank you, Scott France.
Target depth 50 meters. Current depth 4,000. Wind speed
there's my
32.
skid. Okay.
Yeah, Les
Go away.
Watling agrees. That's a parasite. Hey, I'm getting good at guessing.
So for
Next
those of
target
you who are
depth, 133.
watching from home and you're
hours,
looking
get
at
to
your
50 at
feed 2,
18.
your screen 2, and you see it, that the view from the Sirius down to
3
the Deep
local, so
Discover,
3 UTC.
you may be
yeah.
wondering what the bulb is on the tether that looks like a bumblebee body. It seems to behave
This
like
is the
a
current
float.
time.
So
That
this
takes
was
us 2
a
hours from
kind of a
now, takes us to
nerd question, but
3:30.
in fact it is a float to keep the tether from sinking too low.
Watch lead pilot.
Go ahead, pilot.
You had asked earlier, before a few zooms, to describe how we keep track of the vehicles on the seafloor.
And we would love to hear you tell us.
Okay.
Yeah, go ahead.
Is now a good time?
Yep, now's a good time. Yeah, so for those of you who are wondering at home how we know where we are, how we keep track of where we are on the bottom, we are going to have Carl, are you going to do it? Or is
I
Nav going to
think
do it, or is Jim going to do it?
I'll turn it over to our navigator, Richard, to
Richard.
describe how we track
Richard
the
just looked.
how we track the vehicles
That's
on the
because
seafloor.
we like to listen to his British accent.
Exactly.
Okay, so you're going to have to speak up.
How do we track the vehicles on the seafloor?
Can-
At this depth, we don't track them particularly well. We use a system, an ultra-short baseline system, where we, on one of for the Sirius vehicle, we will actually ping down from the ship. That will activate a transponder on the vehicle, which will ping back. And we will estimate the range and bearing from the time it takes to receive that ping. It's slightly different on the main ROV because that actually has a digital signal which when it pings out in one direction, so it should be more accurate from the ROV. Basically it's done to do with time travel through the water, acoustically. So both of the vehicles are monitored all the time by sending out one an electrical and one an acoustical signal from the ship and waiting for their returns to come back. Those returns giving us range and bearing.
Thanks very much, Richard.
You're very welcome.
We put you on the spot. Thank you.
And you performed so well.
It's called the hot seat.
And we love listening to your accent. What accent? Okay, so Richard, you might actually have to do that all over again because we're hearing that whoever was talking was not on the OKEX line.
Oh, yeah. Richard, can you do that again?
Okay, so
on
Richard, that was
OKEX?
your dress rehearsal, so you get to do it all over again.
Now put that mic right close to your
Yeah,
mouth.
but he wasn't on the line. Is he on now, Carl?
Put your mic up to your mouth.
So you have to turn on OKEX there.
Yeah, you can't miss this accent, folks. It's fun.
Okay, can everyone hear me now?
Oh, yeah.
Okay, well, wait, we can hear him, but that's okay. Stand by.
Can you hear him on shore? Our scientists on the chat line.
Okay, so go ahead.
Audio's good.
Okay, go ahead, Richard.
Thanks, Richard. Try again.
Okay, this is your navigator here. A brief description on how we track the two vehicles. We have a system on board the ship. It's called an ultra-short baseline system. It's a LinkQuest system. And what that does, that sends out two different forms of signal. One will be an acoustic pulse down to the Sirius vehicle. When the Sirius vehicle receives that acoustic pulse, it'll recognize it, and it'll transmit back another pulse to the ship. the time taken for those two signals to be sent and receive will give us an estimation of the range, and the processing within the receiver on the ship will give us the bearing. The Sirius has a similar philosophy, only when the acoustic signal is transmitted, we send down a digital signal to the Sirius vehicle. On receiving that digital signal, it will send out an acoustic signal back to the ship. And again, it's the time taken for that acoustic signal to arrive at the ship and for the ship to decode the bearing information, which gives us the position of the two vehicles. The digital signal on the Sirius is a slightly more reliable way of communicating because you've only got one travel path. That's why I think that one is on the main vehicle. My lead pilot may back me up on that one or tell me otherwise.
No, that is true. Yeah, the signal to D2 has about half as much error caused by the sound traveling through the water because it's only a one-way acoustic signal.
This is called an ultra-short baseline communication system. And that's how we communicate.
Thank you so much.
Thanks, Richard.
You're welcome.
Okay, so it looks like we have, what, about 2 hours left?
Mm-hmm.
Maybe a little bit more?
Yeah. Nav, can you tell us how long it's going to take us to get up to the summit? I think that's waypoint 2.
Actually, we should call in another move.
Yeah.
And then do some figuring.
Yeah. Yeah, actually, we're a little bit...
The summit currently is still 170 meters. We've been moving about 30 meters every 30 minutes. So we're going to have to change the plan at the moment if we want to make the up to the full summit, the end of the dive. But I'll just get this next move in, and then we can have a look at it.
Okay, thank you.
Yeah, so we might actually have a little bit more time than we originally thought, because first of all, we're a little bit shallower than what our original plan was.
240, Brian?
We have 4,000.
Let's get that.
Meters. I'll get the
move in, then we can.
Thinking like 40 meters, but
bear with me just a second.
Yeah, let's do a 40 meter.
40 meter?
Yeah, your heading's 247. Seems pretty good.
yeah.
Ah, interesting. Up at the
247?
top of the
please.
screen now, we can see...
Bridge from Nav.
Well, it looks like a more
Fair
steeply
leverage.
dipping
Can we have
wall right in
another
front
move,
of
please?
us.
So I would like a 40 meter range at a bearing of 247 and a speed of 0.1 knots. Good copy, Bridge. Thank you.
Video, let's zoom on this new kind of rock. Try to stay near the interface here. You're free to zoom.
So we're going to take a close look at this outcrop now. See if you can tell if it's any different from the other stuff. The lighter color you see at the bottom is just material from which the surface has peeled off. You can see the black above is probably manganese coating, manganese oxide, and below it is the fresher surface, in other words, the non-covered by manganese surface that has fallen off and maybe made a pile of talus just below it.
Jim, do you copy?
You can see fractures in the surface of the rock and small fractures as well as angling
Jim,
up to the right, down to the left. These are larger joints, big fractures in the mass of the rock that we're looking at.
220 minutes, so...
Right, I'm at a partial zoom now.
Let me just have a look.
Okay. No, that looks
140.
good.
Okay.
Sorry, 2 hours, 20 minutes, yes.
No, sometime after 4.
No,
4:15.
4:15. Yeah.
Sorry, folks, we're having a discussion about when we have to leave the bottom.
That should give us plenty of time.
Right. So it looks like we have almost 3 hours left on the bottom. So about 2 hours,
Wow.
about 2 hours and 45 minutes left.
Plenty of
Okay,
time to make it up to the
yeah.
top.
Make it up to the top.
Which
and
is where we saw a lot more animals up on the top.
yesterday on Stegosaurus.
Yeah, it was shallower though, so
Yeah,
I'm
it was
just
shallower.
really curious to see whether we're going to
It'd be
be
very
seeing
interesting to see.
similar kinds of animals up there.
Right.
Found some incredible sponges yesterday that Shirley and everyone on shore
So
were
now,
just oohing and eyeing over,
For
and
reference, I think in the
they
morning,
were
the
so
navigator
wonderful,
should,
and there were like 4 of them
the,
attached to
to a
a
single
rock.
They should
So
write
of course we had to
time at
collect the
surface
rock
and
that had
time
them
off
on
bottom.
it.
Yeah, these were
Look at the
really,
first
really
page,
small
it's just-
samples. They were
were
probably about,
tiny.
Is
oh,
that
I want
there?
to say
Does it
less than 4
4:15
centimeters
off
long.
Yeah, that's
Some of
true.
them were like 2 centimeters.
Right.
I'd say 2 to
So
4 centimeters.
no way to
Four
pick 'em up
different,
in the manipulator.
Yeah, probably at least 3 different species, maybe
It's
4.
on the first page.
Got them back intact, and they are going to be ready for someone to analyze.
Time to bottom is 142. That should be 2 hours, 22 minutes. Okay.
I don't think it's on there, Carl.
Oh, it's time at
So
50
Deb's saying that the right side
Yeah.
looks like it's really altered.
Time off on.
Correct.
I agree.
At 50 meters for recovery time, 6:30.
Okay, but not the next step. All right.
Deb Blixen, our shore-based geologist, who actually was the geology lead on leg 1 of this 3-leg series of
like
cruises
ships moving.
for the Mariana Trench National Marine Monument, is saying that the rocks on the
When we talk
right
about
side of the
recovery
screen
at
look
6:30,
really
that's on
altered.
board at
I think they
6:30, or-
also
No, that's
have
50 meters at
a
6:30.
partial coating of fine
Okay.
sediment that
50
it's
meters.
just draped
So
over
that-
the
yes,
surface of the rocks.
The 4:15 is
But I
fine.
agree. I think
Yeah.
all these rocks
I was
are
having it on
pretty
bo- okay.
altered.
So the first leg focused mostly on the southern part.
That's right.
Of
The
this-
southern
of the-
half of the Mariana Arc, which stretches for from about 13 degrees north
So
to
the
about
top of this
24
thing is
degrees
actually
north.
at,
Right, and then during the second leg, the mapping
What did it
team
say,
was out,
42,
and
20?
they did.
Yeah.
They
They
conducted
did
some more mapping of,
Right.
areas that had not yet been mapped, and
With modern mapping
yes,
equipment.
Yeah,
exactly,
accurate
and,
is,
and enabled us to refine
contours.
some of our dive targets for this leg.
That's right.
Places you don't know, but certainly I think the top ones are usually correct. You know, this
Yeah, yeah,
is
yeah.
doing all sorts of weird things. So you're underway probably? Gonna start moving in a minute?
Yeah, I think so.
So all of the formation that
there's
we're
no
looking at,
significant
whatever
targets,
it
we
may
can,
be, and we do have a sample of
try
it, so
to-
we'll be able to
try
tell later.
Push the moves closer together.
I'll get a chance to poke at it a bit and find out
See if you
whether
can't
it's
cover a
a
little
rock
ground.
or
Yeah. So it
or
should be,
a
according to this, it should be
consolidated
flattening out.
sediment, partially consolidated sediment, which
And
we would
I
also
actually
call
agree.
a sedimentary rock,
60
but
meters
it
down
is
and
not.
that's 60 meters right
Those
there.
are not
Okay.
igneous,
We're on the right
not
contour,
formed by a
actually.
volcanic eruption. We'll find out what sort of material this is that we've been looking at. And when we get to the top, we can still collect yet one more rock sample. So we can try to do that up closer to the summit of the ridge, and then we can get possibly an indication of the age of the strata. If these are sedimentary materials, we can tell the difference between the ages at the bottom and the top, which is useful for trying to figure out what accumulation rates there are if these are sediments. And if they are igneous rocks, volcanic rocks, we can determine the ages at which they might have been erupted or emplaced underground as intrusive bodies. That's magma that just intrudes through fractures in the crust of the earth. When we get up to the top, if we get igneous rocks, they're very likely to be younger. And we can tell then for how long igneous intrusions, igneous processes may have been going on in this part of the Mariana region. We're situated about, I think it was about 30 or 40 kilometers west of the Mariana Trench axis. And the island arc, so where the volcanoes are, part of the Ring of Fire, situated at 200 kilometers west of the Mariana Trench in about this latitude. So we're looking kind of at the crust in the middle part. All of the crust of the Mariana system from the trench axis to the active volcanic arc, that 200-kilometer wide swath of ocean floor, basically records the history, the entire history of growth of the forearc region over a period of approximately 50 million years, give or take 5 million years. When the
really
subduction first began
around.
at the Mariana Trench area. At that time, about 50 million years ago, the Mariana Trench is not where it is today. The islands that were forming as volcanoes at that time were oriented east-west and were about at the latitude of the equator.
This is what I had before.
And the whole system
So
has
if you shrink the map screen,
changed, has moved
About that
northward and
size.
rotated clockwise into its current position, which is almost north-south. Beautiful outcrops
hauling up a
of
bit,
broken
Pilot.
up rocks
Definitely on the move.
all over the
Copy
scarp
that.
face that
Work
were
our way up.
rising up.
You've got your sonar here to tell when you're perpendicular, and that there's nothing off to your right or left.
Okay.
You can actually maybe... Nav, could you zoom the sonar into 20 meters?
So we started our dive at
Yes, please.
4,422 meters, and we will be reaching our waypoint 2 at the top of the ridge at 4,225
gives you,
meters, approximately.
You know, if you're zoomed in with your camera...
I don't know, Shirley, you want to look at some of those
cameras
white
are
things?
distorted, so you can't quite
Looks
tell
like
when
it's
you're
just
square
more
up.
that same
That gives you your
white
angle pretty well.
Oh, we
Copy
can
that.
zoom in closer and take a look. We can zoom in closer.
Yeah, video, feel free to zoom in a little bit.
Yes, please, pilot. You know, the white things, the potential barnacles.
Copy
Ha
that. We'll see what we can do for you. Get a little closer.
I don't know. This looks
It
like
does
it
look
might be on the
it looks
surface.
It's a lot
different.
easier the closer you are.
Copy.
The more we zoom in, the more I think it's staining on rocks, but maybe not. And there's a little horizontal white thing just to
Sonar
the
looks
left of
pretty
center
good.
at 9 o'clock.
The ridge ahead is
I do.
still fairly ridge-like.
Actually, there are
I love
lots of
zooming
those.
in.
There are lots of those horizontal white things.
Are there?
Yeah.
Okay.
It's just like it's like
Just
rock, but we can take
a
a look.
surface
Let's
of a
take a
rock.
look.
And we will be zooming shortly.
Below lasers or above lasers we're talking?
Let's see.
Just-
It really doesn't matter. It's just pretty much
Yeah,
anywhere.
because I mean, as
where
we're
the
getting
lasers are.
closer, it doesn't look to me like there's anything that's
Yeah, I think it's
Living.
from a distance that looked like it was white. As we get closer to it, it doesn't look like it's that.
Right.
Yeah, but you can zoom in and see what we got in
Yeah,
here.
we can look.
Zooming is fun.
Yeah.
Because then you can see those horizontal. See the horizontal you were talking about, Patty. Okay.
Well, I
so
see
we
a
can take
vertical
look.
thing that has things
So,
sticking off of
okay,
it.
all right, there we
alive there.
go. There's something right above the lasers.
Kind of in the middle
Cover a little
upper.
more video if you
yeah, now it's right in the middle of the
Bringing it a
screen.
little closer here.
So Shirley, what is
be
that?
aware.
I don't know. As soon as we
So-
get close, I can
Well,
take
you realize,
a look
folks
at
at home, you know, there's some moving around because the pilot is
It's
trying to
such still-
hover in the
such
water
still water.
column so we
They
can
usually
get a
give
zoom on
little
this.
tiny
Hydroid?
bursts up and down.
it.
Almost barely moving the stick.
Possibly.
That's
If
a
you do
weird
anything
looking hydroid
continuous,
if
you
it
usually
is.
end up
Possibly.
overshooting.
Looks like
Copy.
something else. Wow. No idea what that is. Anybody on shore know what this
Today's
is?
a lot different than
Tara Luke says
probably the other times last cruise when
biology.
you flew,
Deborah
there's a lot
Glickson
of
says
current.
bio. And Asako says it's a crinoid.
Well, question mark.
Very small
Yes.
crinoid.
Question
Very
mark.
small crinoid.
Question mark. Crinoid.
Well,
is
at
it?
least we saw a critter.
Well,
Okay, Pat, I think I'm going to... got a little bit of time.
Yeah, I'm definitely moving, but you got plenty of time. My camera's
What
at
about
50.
lateraling to the left a little?
We can do that. You can pull out a little bit video. Partial.
A
Lateral
really,
left.
really tiny
You're pretty
crinoid.
far away.
Okay, and I think there might be another one right in the middle
Come
of the
in.
oops.
That was just looked like that one was just like the stalk.
Yeah. Well, there were things fanning out from the bottom
Come
of
in.
it.
Oh, the other one? Oh, okay, I didn't
Mhm.
see that.
Okay.
Oh, not much here.
With the fairly gradual slope and everything stowed away, you don't really
Not
have to
a
worry
lot
about
of food
bumping, 'cause
this
the worst
deep.
that'll happen is
Skid.
We got a
Bump, yeah.
little explanation from Scott France a while ago, and
can
he
be
was
jarring to
saying
the
that,
video
you
if you
know,
bump, but
they're
otherwise
just,
it won't hurt anything.
there's nothing in the water column. The water's really clear down here. Not much for critters to eat.
You can pull out video. Yeah, right there's good.
Yep, I think I was desperate for animals. It's really nice to see the rocks this close, too. The fracturing here is just all over the place.
Ah, that was the real
That was the
reason.
wind's doing.
real reason.
That was the real
Yes.
reason.
Currently, we are at about.
No, I like the
14
critters.
knots.
Yeah,
From-
the wire cam's looking a lot calmer
There was a
than it
little bit
was
of
a couple
discussion
hours
about
ago.
that really tiny animal that we saw. So Asako thought it was a crinoid. Bob
because
Carney said
he's
yes, it's
now
pentaradial. And then
wherever
Allison
we
Miller
are.
from Guam joined us, and she
Hey,
said, "Wow,
hi.
probably
Awesome.
knew."
Top of the day.
Gotcha.
Pilot, I'm still at a partial zoom.
Copy. You can pull all the way out. Video. Thank you.
The currents, yes, are a lot better.
Thank you.
Coming up, pilot.
So our dive
Copy
supervisor
that.
just said the currents are really light, so that's a good thing. Easy to control the ROV.
Yep.
Camera's 64.
Tara, is that you?
Nav, how's the ship move progressing?
Yeah,
We are
Asako Matsumoto.
Nope, it actually wasn't
Okay.
me.
Okay. I heard somebody beep in.
Asako Matsumoto
how
said
long
that
have they been
the
taking? Did you
the
say
thing we
10
looked at was
minutes?
tiny
We've been about
and
12
was
minutes,
branching at the
Okay.
tip. I saw
Yeah.
that too.
Yeah.
Pentagonally, we are told.
Pentaradially.
Excuse me. I keep saying pentagonal because I do crystals, and they have pentagonal
since I'm not
symmetry.
moving much, instead
But
of just
pentaradial
forward,
for
you can
animals.
search, side
For
to
I'll
these,
get it
side.
eventually,
for these
Charlie.
animals, for these
Otherwise, you might
animals
get to the
anyway.
end and then we have to wait
Well,
20
we are
minutes for them.
halfway through the cruise
Copy
on
that.
this dive
See
today,
what we got on the right side.
and I still
there.
haven't got it right.
Oh no, but you're getting there.
I know what a barnacle is now.
But it's
And I know
nice
it
that
has
you're
feet,
a bit ahead,
and
so
you know
all
it's
of a sudden
sticking out.
Right, and
they're
not,
called
and they're not
series.
right. Not
They're not
tentacles.
arms
pops
or
up
not
sonar,
tentacles.
You can just go up there.
Yeah,
Yeah, I was reading about
42 seems
barnacles,
pretty happy to me.
and what they do,
Yeah.
folks, is they attach to whatever it is they're attached to by their, quote, foreheads, unquote, is what I read.
Okay.
But I read that on Wikipedia, so take it with a grain of
See.
salt.
My sonar is showing that I'm slowly moving
So
ahead.
I
Got
asked
a very small white little guy.
Tara
Video, you can try
Harmer
to get in if you
Luke,
want.
since Tara
He's a
teaches
little bit
a
long.
course in
Quick.
deep sea biology, I
Get in
asked
there.
her if maybe she could
He's
talk
little.
to us a little bit about.
Ooh, a rock zoom.
Well, actually, I don't think they were
Yum,
zooming
yum.
on a rock. They were zooming on that thing swimming in the.
That
Yeah.
is an
He's little.
amphipod.
Maybe. Anyway,
Starting to
I
lose
asked
you down
Tara
there.
if
Yeah,
she would
looks like
talk
an
to us a little
amphipod.
bit about
Can you
why
pull it
we
in?
are not seeing so much biology here.
Okay.
Because some of our viewers may be
Okay, why don't you
wondering
center
is
back
you know,
up?
why are
Why
we
no
seeing
critters?
why are we seeing so few things down here? Tara, you want to take a stab at it?
Sure. One thing that's really important about the deep sea is there's a whole lot less food than you find in shallower waters. Because you don't get sunlight this deep, you don't get any photosynthesis, so you don't get any algae, any things that use the sunlight to
Tilting
produce
down
food.
to get you.
The area that we're looking in right now doesn't seem to have any hydrothermal activity or doesn't have any activity that
Coming up.
looks like there's some sort of a food supply for chemosynthetic bacteria.
There might be a
So
spot
we're in
or
areas
something
where
above you,
food is
I'm not
very,
sure.
very scarce. And where food's very
Any
scarce,
issue?
so
If you
is
look
the
at my
biology.
Oh,
You'll
copy that,
find
yeah.
certain
Could just be a rock,
organisms
though.
that,
Reflecting
again,
the
are
light.
able to survive, but typically they have adaptations
Yeah, it looks like a
for
layer of white rock.
not feeding very often, or being able to eat food of various different sizes. So basically, we're not going to
All right,
find a
you're
lot of
looking good again.
biomass this deep
Copy that.
unless
See what we
there's
got on the left side?
something that's providing energy into the system. It takes a really, really long time for particles to fall from this photic zone, that top 200 meters, all the way down here to 4,000 meters or 5,000 meters. And there are a lot of things that can eat it along the way. So very, very little of it actually makes it to the bottom.
Thank you very much.
Okay, Tara, I have a question for you about what we do see on the rocks when we zoom in close. On the surfaces that have the darker coating, which is probably manganese oxide, we see kind of like little filaments that are maybe a centimeter or two long at most, but they're all over the surface. And I've been wondering all cruise long what those things are. Do you have any idea?
Well, it could be a lot of things. It could be the small amount of remains that actually make it this deep. It could have some bacterial association with it. It could be, I don't know, empty egg cases. I don't think it's empty egg cases, but there's a lot of different things that it actually could be.
Okay, video, don't see anything
Do
in
I
here.
think there's microbial activity down here? Yes. But again, bacteria need food as well.
You can come out
So
partial.
there generally has to be some source, whether it's organic or inorganic. But no, I'm not exactly sure what those filaments are.
Yeah.
There are some filaments that are finer than that tend to
You
be
can come full wide video, actually.
filamentous bacteria. Those don't really look like it, but again, as I've mentioned before, bacteria can do a lot of really interesting things, so I would not actually rule it out.
Thank you, Tara. I was wondering if anybody online knows much about the process by which manganese oxide coats surfaces. What I have heard from some of my biology colleagues is that it might be microbially mediated, but I have no clue.
All right, max tension for the last few minutes is 16.
I think I see a biological here coming into view left on my manip cam.
Oh, yeah.
Oh,
there we go.
Hey, biology,
there
folks.
we go.
Some sort of crinoid.
This is a crinoid, I think.
Okay.
What kind of a crinoid? I love
I'll
crinoids.
see what I can do video. I might try to get
Do you
it
like
towed
them more
out,
than
get you a nice
barnacles?
stable shot.
Well, it's a toss-up now because I know the bits
So
and
if you can
pieces
get
of barnacles.
squared up so
Crinoids
both
are a
your
lot
toes
easier
touch.
to. They're a
Okay,
lot
by
less
far the easiest.
complex in
Or
terms of
Get one
their
down and
anatomy.
keep your heading.
Which did you call?
So as I learn more about them, I like them more and more. I'm hopeless. What a nerd. But often, the
Of course,
crinoids
if
are
you get both
prettier.
down and the
They're
angle's
very
slightly
pretty.
wrong,
They
there's
have
not much you can do.
colors,
And they can be
Copy that.
stalked or not stalked.
and we can stalk
Okay,
them
Bridget,
by
just check to the
collecting
bottom.
And
them.
we're
Get back to
stalking
you.
them. So this is not on a stalk, but it's stalked because we're stalking them. Oh my God.
Yeah, we're stalking
We're getting
visually.
punchy.
The currents are dropping
Yep,
off, but
yep,
the
we've
winds
been getting
are
here a
outside
while.
the dominant factor.
And we can
Do you
hear
see an
Chris
issue
talk
with
about
that?
now. He could talk
10
about
degree
prehensile
change.
cirri.
Sounds
He
good.
could.
He could.
Okay, video
I don't think this one
coming
has
in.
them, but he
Will
could.
have no problems for a 10-degree change.
Okay.
Hey, Chris, are you in
But then
the?
again, he might
Go
not.
back. Can you
He
take my
might.
tilt for me?
Okay, if we
Sure
get a
can.
close look,
Yeah.
So you have a little
we can
forward and a
tell.
little
Well,
down.
I won't be that way. I'll go ahead and talk as soon as he's been a little bit
10
further
degrees
so I
port.
can get a better look at him.
Okay.
Okay, I think you're good.
Okay, you're going to be serious and wait till you can be correct. There we go.
I got
So what
both
do you
toes in.
think?
I can't
No,
really turn right.
Allison, if she's
So,
near a telephone, she is an echinoderm specialist.
Really? All right. Let's hear Allison.
You can take off
What do you
the rest of
think?
it and wiggle to the right, maybe. Just the crumbling rocks might allow you to go over.
Has she been on the teleconference line? I'm trying
She
to remember.
has. She has been on. I'm not sure she's on right now. She's at URI. She does not look as if she's signed off, but she might be,
Yeah, I guess.
gazing away,
Is that
doing
sufficient
something in
for
her
this?
office at her desk.
Maybe zoom in.
You want to give a
Rest of
quick
the way.
stat,
Okay,
Chris?
video. Yeah, feel free to do what you want.
Okay, well, so it's hard to say whether those cirri are prehensile, given he's on a rock,
Look at this
so
picture.
straightened out. I would probably guess not, even though they're really long and thin. And I'm not sure why I would say that, but I'm going to go with that's what I'm sticking with. What we're talking about is we're talking about those legs, if you will, or those rods that are coming down from the center of his body that he's perched on
The ship has been in position
the
for about
rock
5
with.
minutes.
And then above that is sort of his central disc or his calyx, which all the arms are coming out of. And so I think I counted 10 arms. And I don't really see anything very distinctive that would go further than it's a feather star.
So about,
It's
I think I could go 10 degrees to starboard, maybe 250 to center
suborder.
up
I
on
think
sonar.
I said that right.
So I think we could do
And
keep
could be
another
something
40
in
that
the
way, maybe as soon as this
Anodontidae,
season's
but
over.
I really don't know.
Chris, I
Sounds good to
see
me, Cobalt.
those little white blobs on some of the arms of the tentacles. We saw that yesterday, and I think Scott said those might be egg cases.
gonadal tissue.
Gonadal tissue, yeah. Which would suggest, said Scott, who is now chopping up his dinner, that this particular fella might be mature enough to have and be getting enough food. Actually,
Yeah, well, it's doing well enough. It's doing well enough.
That's
it's
what I was trying to
it's
say.
doing well. It's healthy enough so that it actually is healthy
Physically
enough,
capable of
healthy enough
procreate.
to be reproductive.
Thank you, video.
Yeah, that's a pretty healthy crinoid. I don't see any problem
Give your
with
camera
them.
back.
Got pretty long arms. It's
So
definitely, I would assume, an adult.
As you're taking off, make
Pretty
sure you
large.
know where it is.
So those could very well be the reproductive
You're trying to
organs
zoom
on
on
the thing. I
something, if it's
just don't know
At a
crinoids
different
well
angle
enough
than you're
to
used
say
to
for
when you
sure
push
if that's
down,
what we're
you'll
looking
actually
at.
get closer.
Copy
So I
that.
usually look down to my skids and then tilt up until I'm happy.
Okay. All right, I'm gonna come up here.
Yeah. So I think I've about slowed, maybe even almost stopped from that last move, and we just calling in a new one.
True, it does.
So you mentioned it's 10 degrees to starboard. You mean, so we're up to 257?
Well, right now I'm at 240, so I was going to
Lots
move over
of
to
talus on
250.
this slope that we're
250,
headed for
okay.
on the right-hand side. Big boulders that have slid down
I'll wait
the
for you to
slope
confirm.
from above.
Okay.
These are quite large. Wonder if any of them carried down any animals. I kind of don't think so.
Yeah, that looks good. It agrees with the terrain.
Call
We're
it
good?
in. 40 meters, 250.
Sounds good.
Bridge, this is nav. Could I have another move, please? And this time I have a range of 40 meters, a bearing of 250, and a speed of 0.1 knots.
All right, I'm looking down at you, 70 degrees.
Coming out.
Sonar looks pretty clear.
Good copy, Bridge. Thank you.
Ooh, yeah. There seems to be an animal maybe over on the right. Is that a fish?
Lighter in color.
Lighter in color, but it could be a rock.
Yes.
I don't know.
We get closer.
Looks like a rock. Yeah, probably a rock. It's kind of angular.
Everything looks like a rock.
Yeah, I think it's a
Yeah.
rock.
Rock confirmed.
Definitely it's a rock. Thank you. Thank you, pilot, for that confirmation.
No problem.
Ooh, nice layers of rock at the top of the screen. 227 layer of rock. Yeah, layers of rock covered over with a finer layer of, I'm guessing, clay. And then there's a layer of blue clay, and then there's a layer of yellow clay, if it's clay, and a layer of white on top of everything.
The ship's door is now open for the next 10 minutes. I say again, the ship's door is now open.
Yep. As I pull up, the slope recedes, so seems to be a pretty steady
Wow.
incline.